Hello everyone, welcome to the channel Microbial World. Today let's learn about prokaryotic transcription. Prokaryotic transcription is the process by which genetic information encoded in DNA is transcribed into RNA in prokaryotic cells. Transcription is the first step in gene expression and it involves the synthesis of RNA from a DNA template. In prokaryotic cells, transcription occurs in the cytoplasm and it is carried out by a single RNA polymerase enzyme. RNA polymerase is a multi-subunit enzyme complex, which means it is composed of several subunits, each with specific functions. In prokaryotes, the core RNA polymerase enzyme consists of 5 subunits, 2-alpha, beta, beta-dash and omega. The alpha subunit is involved in assembly and stability. Beta and beta-dash subunits form the catalytic core responsible for RNA synthesis. Omega subunit plays the regulatory role. RNA polymerase holoenzyme is a complex formed by the association of RNA polymerase core enzyme with a sigma factor. Sigma factor is a regulatory protein. It facilitates the accurate positioning of RNA polymerase at the transcription start site. Transcription involves 3 steps: initiation, elongation and termination. Initiation is the process by which RNA polymerase binds to a specific DNA sequence called the promoter and initiate the synthesis of RNA from a DNA template. Let's discuss the steps involved in prokaryotic transcription initiation in detail. The first step is recognition of the promoter. RNA polymerase recognizes and binds to a specific DNA sequence called the promoter and initiate the transcription process. The promoter contains 2 essential regions, -10 region and -35 region. -10 region is also called Pribno box. It is a conserved DNA sequence located 10 bases upstream of the transcription start site. It has a consensus sequence of TAT-AAT. -35 region is another conserved DNA sequence located 35 bases upstream of the transcription start site. It has a consensus sequence of TTGACA. The interaction between RNA polymerase and the promoter is facilitated by a subunit of RNA polymerase called the sigma factor, which recognizes and binds to the promoter region. Formation of the transcription initiation complex. Once the RNA polymerase is bound to the promoter, it unwinds a small portion of the DNA double helix to expose the template strand. This unwound region of DNA is referred to as transcription bubble. The unwinding of DNA allows RNA polymerase to begin synthesizing RNA using the template strand as a guide. So the initiation complex consists of RNA polymerase, sigma factor and promoter DNA. Initiation of RNA synthesis. RNA polymerase initiates RNA synthesis by incorporating ribonucleoside triphosphates, RNTPs, into the growing RNA chain. The RNA molecule is synthesized in 5' to 3' direction, complementary to the DNA template strand, which is in 3' to 5' direction. RNA polymerase moves along the DNA template, synthesizing RNA until it reaches the end of the gene or encounters a termination signal. Release of the sigma factor. After RNA polymerase has initiated transcription, the sigma factor is released from the transcription initiation complex. This allows RNA polymerase to make transition from the initiation phase to the elongation phase of transcription, where it continues to synthesize RNA along the DNA template. Elongation. Prokaryotic transcription elongation is the phase during which RNA polymerase moves along the DNA template, synthesizing RNA in 5' to 3' direction. So let's see what happens during the elongation process. RNA synthesis. So the RNA polymerase moves along the DNA template, reaches the template strand in 3' to 5' direction and synthesizes RNA in 5' to 3' direction. It catalyzes the formation of phosphodiester bonds between the ribonucleotides, adding ribonucleoside triphosphates to the 3' end of the growing chain. DNA unwinding and rewinding. As the RNA polymerase moves along the DNA template, it unwinds the DNA ahead of the transcription bubble and rewinds the DNA behind it. So the unwinding allows the RNA polymerase to synthesize RNA and the rewinding restores the DNA double helix structure. Proofreading and error correction. During elongation, RNA polymerase has a built-in proofreading mechanism to maintain the accuracy of RNA synthesis. If an incorrect ribonucleotide is added to the growing RNA chain, RNA polymerase can backtrack and remove the incorrect nucleotide before continuing with RNA synthesis. Transcription speed and processivity. RNA polymerase can transcribe genes at speed ranging from a few hundred to over 1,000 nucleotides per minute. RNA polymerase has high processivity, meaning it can transcribe long stretches of DNA without dissociating from the template. So that's all for today. Thank you for watching. Do subscribe the channel for more informative videos.